Welcome to the debate. Imagine mapping a sprawling city, right? You're cataloging every single building, every factory, every road.
Only to realize later that over half the city is just a ghost town.
Exactly, a ghost town filled with dead residents. And that's the exact situation we are facing right now in microbiome science. Our most fundamental maps might actually be lying to us.
It really is a profound realization. I mean, we are looking at this massive fundamental transition in how we understand complex biological systems.
Yeah, we are.
We're moving from statically cataloging what a system is capable of, to dynamically measuring its actual behavior.
Right, and that whole discourse is rooted in the recent comparative literature on next-generation sequencing. Specifically, we're analyzing the conceptual divide between DNA-based and RNA-based profiling.
Which is a massive divide right now.
It really is. So the central philosophical and mechanistic question we are debating today is this. Is a microbiome defined by what it can do or by what is actively doing right now under the pressure of its environment?
Exactly.
And I represent the position that shotgun metagenomics, so sequencing the DNA, provides the stable, reproducible, and foundational blueprint of functional capacity that is just, well, completely necessary for rigorous science.
And I represent the contrasting position, which is that metatranscriptomics, sequencing the RNA, captures the real-time dynamic expression that actually drives pathology and environmental adaptation. So genomic potential is largely insufficient on its own.
All right, so to understand why the DNA blueprint is so critical, we really have to recognize that metagenomics captures the enduring functional architecture of a microbial community.
Enduring being the operative word for you there.
Right, because biology is, at its absolute core, fundamentally constrained by encoded capability. I mean, a microbial community simply cannot express genes it does not possess.
Sure, obviously.
So DNA gives us a chemically stable, highly reproducible measure of what is physically possible. It allows us to escape the transient, volatile noise of just a snapshot in time. When you sequence the DNA, you are mapping the permanent infrastructure.
See, I come at it from a totally different way. That infrastructure, that genomic potential you're talking about, is biologically misleading
How so?
because the vast majority of microbial genes are conditionally regulated. My core argument here is that a genome is not a phenotype.
Okay, but
You have thousands of genes sitting there completely dormant until a specific environmental stressor triggers them. Real-time, behavior-driven by dynamic transcription in response to, say, a host's stress level or what they just ate. That is what actually determines host outcomes and drives disease.
you need the genome to even know what's possible.
But relying purely on DNA is like cataloging all the books in a massive town library to understand what the town is actively thinking about without ever actually checking to see which books are being checked out and read.
Okay, that library analogy is clever, but I think it underestimates the sheer power of having the complete catalog.
Do you really need the whole catalog if nobody's reading half the books?
You do. Because the fault line here is really about requiring stable, large-scale reproducibility versus demanding high-resolution but potentially chaotic accuracy. Just look at the scale of what metagenomics has achieved with GWAS, genome-wide association studies.
Right, the big population studies.
Exactly. We are talking about massive data sets, like the Hunt study and the Swedish cohorts comprising over 28,000 individuals. These aren't just small, noisy samples. They prove that the DNA metagenome is a stable, heritable blueprint deeply intertwined with host genetics.
I won't deny the elegance of the data from those cohorts. It's beautiful foundational work. But we have to look really closely at what those statistical links actually mean in practice.
They mean enduring biological states. We see incredibly robust genetic links. Take the LCT locus, for example.
The milk sugar digestion gene.
Right, the host gene region that dictates whether a human can digest dietary milk sugar. That specific host gene profoundly and consistently shapes the abundance of bifidobacterium in the gut.
Sure.
Or look at the ABO blood group locus, which determines your blood type. It literally physically shapes mucin-degrading bacteria in the gut because it fundamentally alters the mucosal landscape they live on.
I'm familiar with the pathways, yes.
And then you have the HLA-DQ-B1 variants, which are directly tied to the risk of developing type 1 diabetes. And they do so through highly specific microbial abundance modules. These are replicated massive signals. Going back to our city analogy, the host genetics lay the bedrock, and the microbial DNA maps the permanent roads built on top of it.
It is a compelling argument, but have you considered the actual mathematical weight behind those genetic associations?
What do you mean? They're highly significant.
Significant, yes, but when you calculate the overall heritability estimates for microbial traits, meaning how much of the microbiome's makeup is actually inherited from those host genetics, it hovers at a mere 1.9% to 8.1%.
It is still a measurable foundational constraint, though.
A constraint, sure, but one that explains less than 10% of what is actually happening in the microbiome. That leaves over 90% of the variance driven purely by the environment.
But the environment acts on the foundation.
Right. But the traffic on those permanent roads, like dietary shifts, inflammation, xenobiotics, that completely dominates the active microbiome. RNA abundance shifts rapidly to reflect this traffic in real time.
But that shift is...
If you are only looking at the roads, you completely miss the fact that a chemical spill just shut down the highway. The RNA tells you exactly how the community is responding to that immediate life or death environmental pressure.
Okay, I see why you think that, but let me give you a different perspective. If RNA shifts so rapidly, if it changes drastically based on basic circadian rhythms, or a single meal, or even just the few minutes it takes for a nurse to handle the sample in the clinic, aren't you really just measuring transient noise?
It's not noise, though.
It is, rather than a durable biological state. How can you possibly discover reliable, reproducible biomarkers in that kind of chaos?
Because it isn't chaos or noise. It is the precise biological response at the moment of physiological relevance.
I'm not convinced by that line of reasoning, mostly because you are treating the RNA as if it operates completely independently of the underlying genome.
I'm not saying it's independent, but it's decoupled.
But DNA and RNA are mathematically fundamentally linked. Look at the men's lifestyle validation study. They demonstrated that 41% of the variation in microbial gene expression is purely dictated by the physical copy number in the metagenome.
Could you clarify what you mean by physical copy number for a moment, just to be precise?
Sure, if a specific bacterium replicates, its population doubles, right?
Right.
So now you have double the DNA for its specific metabolic pathways. Because there is twice as much DNA, there will naturally be more baseline RNA transcripts being produced, even if the actual per-cell rate of transcription hasn't changed at all.
Okay, yes, I see what you're saying.
By the DNA is a highly accurate predictor of community behavior. You don't always need the RNA to know the pathway is there and functioning.
See, I'm sorry, but I just don't buy that. Let me tell you why. You are quoting that 41% figure as a victory for DNA.
It's a huge chunk of variance.
But that means 59% of the variation is completely decoupled from genomic abundance. That 59% represents active cellular regulation, host microbe signaling, and immediate metabolic adaptation.
But the 41% is the baseline.
And to show you why that decoupling is so critical, I want to bring up the phenomenon of functional redundancy, specifically looking at the colitis paradox.
All right, let's hear it.
So researchers used a dextrin sodium sulfate, or a DSS-induced acute colitis mouse model. Basically, they introduce a chemical that causes severe acute intestinal inflammation.
Right, a massive physiological event. The tissue is heavily damaged.
Exactly. The gut is highly inflamed. When researchers ran the DNA-level shotgun metagenomics on these inflamed animals, they successfully detected taxonomic shifts. The broad category of firmicutes went down, and proteobacteria went up.
Which is the expected inflammatory profile.
Right. But when they tried to infer the functional metabolic pathways from that DNA, the actual chemical processes happening, the DNA-level analysis yielded zero statistically significant differences. Zero.
Well, that happens because of functional redundancy. Let me jump in and explain the mechanics of that.
Go ahead.
If the main bakery in our city burns down, but 3 smaller corner cafes suddenly buy ovens and start baking bread, the city still has the exact same bread-making capacity on paper.
Right.
Different bacteria have overlapping, redundant metabolic pathways. Under stress, one bacterial population drops, another rises, and the DNA copy number for those shared pathways balances out.
Exactly. At the DNA level, they compensate for each other. So the total metagenomic potential looked completely static.
Because the capacity was preserved.
But the DNA profile was utterly blind to severe active pathophysiology. Now contrast that with the Metatranscriptomics, the RNA results from the exact same model.
What did the RNA show?
The RNA captured massive, real-time metabolic perturbations. It showed the active upregulation of GTP-binding proteins, which mechanically amplify immune cascades in the gut.
Okay, that is significant. It showed the activation of signal peptidase 2, an enzyme that drives virulence. It even captured the upregulation of an anti-inflammatory enzyme called L-asparaginase, revealing that the bacteria were actively trying to suppress the host's immune cell activation.
Fascinating.
None of those active critical survival mechanisms were visible in the DNA. The DNA said the city was perfectly fine. The RNA showed the city was quite literally on fire and mapped out exactly who was fighting the flames.
That is an incredibly powerful mechanism, I'll give you that. But everything we've talked about so far assumes a neat, living ecosystem like the human gut.
Where are you going with this?
What happens when we look at an environment that is full of death, like soil?
Oh, I'm so glad you brought that up, because this is where DNA profiling stops being merely blind and starts creating absolute illusions. Up to 55% of total extracted soil DNA can be what we call relic DNA.
Right, necromass, the genetic remnants of dead cells.
Yes, it is dead, lysed microbial cells whose DNA is physically shielded from degrading by soil mineralogy. It just binds to the dirt and sits there completely inert for months or even years.
And the sequencing picks it up?
Traditional metagenomics extracts this indiscriminately alongside the living cells. This falsely inflates biodiversity estimates by massive margins. It completely misrepresents nutrient cycling.
You're thinking of the nitrogen studies?
Look at the LAMPS trials, which studied nitrogen fertilization in agricultural soils. When they compared the data, the RNA libraries showed a 7-fold higher active nitrogen cycling bacterial community than the DNA libraries. By relying on DNA, you severely underestimate key biogeochemical functions because you are literally swimming in a sea of dead genetic material.
That's an interesting point, though. I would frame it a bit differently. I can see the relic DNA point in soil matrices. It's a known physical artifact of that specific environment. But you don't even have to look at soil to see this. We see it in humans, too.
Wait, how so?
In the human gut, there is a bacterium called Dialister invisus. It is highly abundant in fecal DNA but completely transcriptionally dormant. It's essentially asleep.
Right.
Or look at oral microbes that get swallowed and somehow survive the acid of the stomach. They arrive in the lower gastrointestinal tract as passive, non-functional genetic relics. They show up perfectly on your metagenomic map, but they aren't interacting with the host at all.
So wait, so you agree that relying purely on DNA is inherently flawed?
I agree that dormancy exists, but I am going to push back incredibly hard on the idea that RNA is the perfect solution, because we need to talk about the immense physical and computational fragility of metatranscriptomics.
Fragility?
Yes, you portray RNA as this clear, pristine window in active biology, but getting that window is a literal extraction nightmare.
It's technically demanding, sure.
Working with RNA isn't like reading a stable blueprint. It's like trying to read a self-destructing message. The moment the cell opens the RNA to build a protein, enzymes are already rushing in to tear the RNA apart. It degrades in minutes.
Which is why we flash freeze.
But it requires extreme chemical preservation right at the precise millisecond of sampling.
It absolutely requires rigorous precision in the lab. I won't deny that.
It requires near perfection, and even if you capture it perfectly, up to 90% of the sample is dominated by useless ribosomal RNA, or rRNA.
The structural material, yes.
Right, the structural material of the cell, not the messenger RNA that actually tells us about behavior. You have to physically deplete that RNA in the lab, which introduces massive chemical bias into your sample.
Well,
When you use chemicals to pull out the ribosomal RNA, you accidentally pull out some of the messenger RNA with it, altering the ratios. What you finally sequence is already a heavily distorted reflection of biological reality.
But we have robust enzymatic digestion protocols to handle RNA depletion now, which minimize that bias significantly.
But they don't eliminate it. And we cannot ignore the math here. The statistics of metatranscriptomics are an absolute minefield. You are dealing with massive structural and technical zero inflation, dropouts everywhere.
That's a solvable problem.
You sequence a sample, and a critical gene shows zero expression. Is it actually repressed by the bacteria, or did the sequencer just fail to pick up a low-abundance transcript because the RNA degraded too fast?
That is primarily a sequencing depth issue. You just have to sequence deeper.
And that brings me to scalability. Sequencing deeper costs money. Metatranscriptomics costs roughly $237 per sample, compared to about $86 for DNA.
The cost is dropping, though.
When we were trying to do population-scale biobanking with thousands of subjects, RNA is prohibitively expensive and technically unstable. And look, I see why you think that focusing on active expression is better, but let me give you a different perspective on why ignoring the DNA is downright dangerous.
Okay, why is it dangerous?
DNA successfully captures rare but clinically vital elements that RNA completely misses, simply because they aren't firing at the exact microsecond you drew the sample.
Ah, you are talking about latent capacity.
Yes, exactly. Dormant antibiotic resistance genes, latent virulence genes, prophages, viruses hiding inside the bacterial genome, waiting to break out.
Which are important, yes?
If you only look at the RNA, you might look at a pathogen and say, oh, it's harmless, no virulence genes are currently active. Then the patient's internal environment changes slightly, the gene activates, and the patient crashes. Ignoring latent capacity is how you miss the time bomb before it goes off. Biology is ultimately constrained by encoded capability. Transcription is transient, genomes are enduring.
The fragility of RNA is a formidable challenge, I certainly won't deny that. And you are entirely correct about the statistical complexity, particularly the confounding of transcript abundance by the underlying DNA copy number.
Right. Going back to what we discussed earlier, if a bug replicates, it has more DNA, generating more baseline transcripts, confusing the signal.
Yes. If you evaluate metatranscriptomic data in complete isolation, you will mistakenly identify those basic DNA-level abundance shifts as differentially expressed genes. It creates a very high false positive rate.
Exactly. But that is exactly why the field is moving toward a multi-omics resolution. The solution isn't to retreat to the safety in the blind spots of DNA. The solution is statistical integration. We have to sequence matched DNA and RNA from the exact same physical sample.
So you're talking about joint conditional modeling here?
Precisely. We use emerging paradigms like IMIC, that stands for the integration of metatranscriptomes into community genome-scale metabolic models.
It's a mouthful. It sounds dense, yeah, but the concept is actually straightforward. By mathematically modeling the observed RNA counts as a direct function of the paired metagenomic DNA copy number, we explicitly decouple true transcriptional regulation from simple structural genomic variation.
So you're finding the baseline.
Right. We are basically looking at a ratio, how much RNA is produced per unit of DNA. We subtract the DNA baseline to reveal the true RNA regulatory action.
Hold on, let's break that down for a second for the sake of clarity for everyone. When we talk about sequencing data, we have to acknowledge that next-generation sequencing is compositional. It's trapped in what statisticians call a simplex constraint.
Which is a huge headache.
To put that simply, because we sequence percentages of a whole, everything has to sum to exactly 100%. So if one specific bacteria rapidly multiplies, its percentage goes up, which mathematically forces the relative percentage of every other bacteria to artificially drop, even if their actual numbers haven't changed at all. It is a math illusion.
It is, and it creates massive spurious correlations if you don't mathematically correct for it.
Exactly. That's why we absolutely require frameworks like the centered log ratio or CLR transformations.
Yes, CLR is essential.
That is a mathematical technique that pulls the data out of percentages and maps it into standard Euclidean space. That way, variables can move independently without forcing other variables down. And we need zero-inflated models, like Tweety distributions.
To handle the dropouts.
Right. The Tweety distribution is a statistical tool specifically designed to handle data that is absolutely full of zeros, so we don't accidentally confuse a missing read from the sequencer with a bacteria that is actually dead.
Exactly. Both layers, the stable DNA and the dynamic RNA, are fundamentally necessary if we want to stop treating physical abundance shifts as false positive expression changes. It is the only way to build models that actually predict metabolic flux and biological reality.
I agree. So, as we wrap up this discussion, I want to summarize where the evidence leaves us regarding the DNA blueprint. Despite the complexities, metagenomics retains profound, enduring value because it provides the essential map of functional potential.
It maps the territory, sure.
It defines the hard biological boundaries of what an ecosystem is physically capable of achieving. It maps the deep, heritable traits shaped by host genetics, and importantly, it catalogs the rare, latent capacities like antibiotic resistance that we simply cannot afford to ignore just because they are momentarily quiet. The genome is the foundational architecture upon which all biology is built.
And from my perspective, the metatranscriptome represents the crucial execution of that architecture. Having a blueprint doesn't mean the building has been constructed, right? And having a metabolic pathway and a genome doesn't mean it's actively being used.
Fair point.
Metatranscriptomics captures the dynamic, real-time responses to the environment, the inflammation, the dietary shifts, the stress that actually drive biological and clinical outcomes. As the colitis paradox and the soil relic DNA problem showed us, relying on potential alone creates severe blind spots that obscure true physiopathology and environmental reality.
It seems the emerging scientific consensus is that neither omics layer is truly sufficient in isolation.
They are inextricably complementary. Potential constrains behavior, but expression determines phenotype.
Beautifully put. We'll leave it to our listeners to ponder how they view complex biological systems, whether through the enduring lens of encoded capacity or the fluid, responsive reality of active execution. There is much more to explore in the underlying research.
Indeed, there is.
Think back to our city. You can study the concrete layout to know exactly where the roads are built, or you can track the headlights to see how the city lives, adapts, and breathes. The ultimate truth of the system requires both. We'll see you next time.